Hi viewers, this is J. Swami, Assistant Professor of Zoology. In this class, we are going to discuss animal bioreactors, concepts and applications. Under this topic, we are going to discuss the introduction to the bioreactors, definition, animal bioreactors and types, recombinant proteins and their expression, animal tissues as the bioreactors, mammal animal bioreactors, rabbit, which is an excellent model as the bioreactor, and the animal bioreactors and their applications. Let us start with the bioreactors. Bioreactors are defined as an apparatus for growing organisms like yeast, bacteria, animal cells under controlled conditions. They are used in industrial processes to produce pharmaceuticals, vaccines, or antibodies. They are also used to convert raw material into useful byproducts, such as in the bioconversion of corn into ethanol. A bioreactor provides the optimal conditions for achieving the desired product by providing optimum growth conditions. Generally, bioreactors are vessels in which raw materials are biologically converted into specific products using microbes, plant, or animal cells. The first microbial bioreactors, in particular Escherichia coli and Saccharomyces cerevisiae, were found to be satisfactory for the production of simple polypeptides such as insulin, human growth hormone. However, microbial bioreactors were found to be unsuitable for proteins with complex post-translational modifications or intricate folding requirements such as the coagulation factors or monoclonal antibodies. Hence, the idea is evolved to make use of animals as the bioreactors to produce various proteins and other components along with their products like milk, eggs, meat, etc. The animal bioreactors are defined as the animals that are used for the production of recombinant proteins, which are highly useful for the welfare of human beings, such as proteins used in the treatment of certain diseases, to reduce the transplant rejection, to meet the need of nutritious food material, etc. For this, we have to produce genetically engineered organisms or transgenic animals. The proteins that are produced should be biologically active and should be formed after proper post-translational modifications. Work on transgenic expression systems using animals began in the early 1980s, primarily as a way of improving the genetic characteristics of livestock. Transgenic animals acquire genetic material through human intervention rather than through normal sexual reproduction. The hope was to accomplish with microinjection of functional genes into ova, what would otherwise take years with traditional breeding programs. In an early experiment, a super mouse was created when a target gene for growth hormone was injected into and expressed by the genome of a particular mouse. And there are specialized research animals are there. Aside from application designed to improve characteristics of a particular species or to create specialized research animals. Transgenic technology is also achieving increasing success as an alternative to producing proteins in cell culture and microbial systems. The goal of such work is to produce large quantities of recombinant proteins in the milk or plasma of transgenic mammals or in the eggs of transgenic hens. Clinical trials are successfully completed. Companies like GTC Biotherapeutics, Europe, GTC Therapeutics, Europe. Here, this company reviewed for market authorization in Europe for a particular recombinant protein which is called as antithrombin. It's called as a antithrombin, ATRYN. It is a recombinant form of human antithrombin expressed in the milk of transgenic goats. And the production of recombinant proteins is one of the major success of animal biotechnology. And here milk, egg white, blood, urine, seminal plasma and silkworm cocoons of the transgenic animals, they are the source of the recombinant proteins. They are the main source of the recombinant proteins at an industrial scale, large scale of production. The generation of transgenic farm animals has become recently easier, mainly with the technique of animal cloning using transfected somatic cells as a nuclear donor. And then numerous experiments carried out for the last 25 years have shown that the expression of the transgene is predictable only in a limited extent. Next, the biotechnology industries, they are generating the transgenic animals. So the gene with the desirable characters is the transgene, and insertion of this transgene into an embryo, otherwise into an ovum, and production of the recombinant animals is nothing but the transgenesis. And the newly formed organisms with two kinds of genome, its own genome and the desired gene of our interest, they are called as the transgenic animals. We follow different methods for the process of transgenesis, mainly microinjection method, embryonic stem cell method, retroviral method, and the SCNT, somatic cell nuclear transfer method. Here there are different platforms which are used as the animal bioreactors. Biotechnology industries is experiencing an extremely low manufacturing efficiency for recombinant therapeutic proteins, especially for proteins with post-translational modifications such as acetylation. There are some problems in many existing recombinant protein expression systems. For example, bacteria usually lack the post-translational modifications required by many proteins. Unicellular eukaryotic species like yeast, filamentous fungi, and unicellular algae often yield foreign proteins with undesirable properties or yield products with no activity caused by their inability to follow human patterns of protein processing. Insect systems have their own unique glycosylation patterns. Mammalian cell culture systems are expensive to maintain and difficult to scale up. However, compared with these systems, transgenic animal bioreactors were able to produce complex biologically active pharmaceutical proteins in an efficient and economic way. There are many protein production platforms for transgenic animal bioreactors. For instance, foreign proteins produced by mammary glands are secreted into milk. By liver and other tissues, they secrete into blood, and by the bladder into urine, by ovary into egg white, and by seminal vesicle into semen. These are the better platforms which are the tissues of organisms which are used as the sites of animal bioreactors. Let us see one by one. Currently, mammary gland is the best available animal bioreactor. However, it is limited by the discontinuous nature of the lactation cycle, and a certain number of failures have occurred for various reasons, such as purely technical difficulties in generating transgenic animals, lack of knowledge about protein maturation or secretion in milk, adverse effects of certain proteins on animals' health, and reabsorption of some of the proteins. The next one is the liver and other tissues can secrete recombinant proteins into blood. However, blood is not an ideal fluid for protein production because it collects secretions from many other non-target tissues, and many proteins are poorly stable in serum. In addition, blood collection is invasive, and some proteins may have serious impact on the health of the animals, which makes the system impractical. Followed by, bladder is an alternative system for producing foreign proteins. Urine is an abundant biological fluid which can be collected more easily and much earlier than the production of the milk of a organism. The bladder epithelium secrete proteins but at very low rate. Then coming to the ovary, especially ovary in hen is another ideal animal bioreactor. A single hen can produce up to 330 eggs per year, and egg white is also an abundant fluid naturally containing 4 grams of protein per egg. 4 grams of protein per egg. It has not been adopted so far because it is technically difficult to generate transgenic birds. Coming to the vesicular gland, otherwise a seminal vesicle. Vesicular gland has been reported to be a promising animal bioreactor. It secretes proteins into seminal plasma. Seminal plasma is relatively abundant biological fluid in some species such as swine, and it can also be easily collected. However, to date, only seminal vesicle in male accessory sex glands has been reported to produce foreign proteins in the semen at laboratory level. Next, come to the epididymis. Epididymis has very strong secretion capacity and can secrete at least several hundred kinds of proteins. The naturally secreted proteins by epididymal epithelia are released into epididymal lumen and finally become the constituents of semen. Mammalian epididymis can be used as a bioreactor to produce pharmaceutical proteins. Because the biological and genetic background is relatively clear. Why we use epididymis as the bioreactor? The strong reasons are the biological and genetic background is relatively clear. Secreted proteins have several post-translational modifications. Certain epididymal proteins in high content in semen Proteins secreted by epididymis can be stored. Proteins from semen can be easily isolated. Production of foreign proteins using epididymis may be achieved at low cost. Transgenic animal bioreactors are attractive platforms to address the growing needs for therapeutic recombinant proteins. Therapeutic recombinant proteins. The ability of transgenic animals to produce complex, biologically active proteins in an efficient and economic manner is superior to those of other organisms. Another important consideration is cost. Building a large scale of about 1000-liter bioreactor, let us assume, manufacturing facility for mammalian cell takes 3 to 5 years and costs of about 250 to 500 million US dollars, whereas a transgenic farm with a single purification facility should not cost more than 80 million US dollars and would most likely cost less. The various mammals used as bioreactors are rabbits, pigs, sheep, goats, and cows. Each of these species have advantages and the drawbacks. Most of the times, the transgenic animal bioreactors, they are the active platforms to produce different kinds of recombinant proteins. Let us start with the cow. Transgenic cow as animal bioreactors. The prospect of obtaining a large amounts of milk produced by dairy cows made them early candidates for studies into transgenic production. Dairy cattle produce 23 grams of protein per kg body weight during peak lactation. The transgenic cow can produce blood clotting factors number 8, blood clotting factor 9, protein C, and the antithrombin. The Dutch company working with developing of transgenic cows with the creation of Herman the Bull, designed to breed progeny that produce lactoferrin. Hermatech company is working on production of human polyclonal antibodies in transgenic bovine plasma, so that the cow works as a efficient animal bioreactor. This is the Herman the Bull, which can produce lactoferrin in its progeny. Then come to the goats. Transgenic goat as animal bioreactor. Goats are smaller than cattle and also produce a large amount of milk in short time. Goats can produce alpha 1 proteinase inhibitor, monoclonal antibodies, Ig fusion proteins, recombinant human antithrombin 3, and TPA, tissue plasminogen activator. TPA is the tissue plasminogen activator. And coming to the next one, transgenic mice as animal bioreactors. Mice may yield more amount of milk compared to larger species. They are still powerful little bioreactors. Peptides derived from anti-neoplastic urinary protein were shown to reduce tumor by 70% in nude mice and human cervical cancer cells. Other research work with transgenic mice includes expression of malaria protein for possible varieties, monoclonal antibodies and Ig fusion proteins, alpha 1 proteinase inhibitor, antithrombin, angiogenin, cystic fibrosis transmembrane regulator, factor 10 blood clotting factor 10, glutamic acid decarboxylase, glucocerebrosidase. HGH is the human growth hormone, proinsulin, prolactin, and fibrinogen. These are the possible recombinant proteins to be produced by mice, which is a most efficient animal bioreactor. And come to the transgenic pig as the animal bioreactor. Pigs are mostly used for xenotransplantation research, but transgenic pigs have produced human hemoglobin and human protein C. Pigs are proving to be valuable research models for retinitis pigmentosa and other eye diseases. Future applications may benefit from similarities between human and pig digestive and cardiovascular systems. And let us see sheep, transgenic sheep as the animal bioreactors. When coli A1 gene from connective tissue cells, that is a fibroblast, was combined with a vector and fused with enucleated sheep eggs, two lambs secreted milk containing type 1 collagen of 650 micrograms per ml. Fibroblasts secrete type 1 collagen, the absence of which in human causes osteogenesis imperfecta. Osteogenesis imperfecta. And transgenic rabbit as the animal bioreactor. Rabbits appear to be attractive candidates as transgenic animals. They are cost effective to raise. They reach sexual maturity after 5 to 6 months. Average 8 offspring per pregnancy and can produce up to 40 embryos following superovulation. Their milk has high protein content and they can produce up to 250 ml of milk per day. Rabbit milk naturally contains 2.5-fold as much protein as sheep milk and 4.5 to 4.8 folds that of goat. Expression levels of transgenic protein can be as high as 20 grams per liter. For small and medium sized facilities, for small and medium sized facilities, the rabbit system is ideal to produce up to 50 kg of protein per year. Thus, the transgenic rabbit system is a lower cost alternative, primarily because rabbits are smaller and less expensive to maintain. The livestock. Specific pathogen-free rabbits are available and free of infectious agents. There has been no known serious disease transmission to humans from the rabbit, which makes the rabbit safer than other dairy livestock and poultry. Therefore, the system of transgenic rabbit is safe to produce therapeutic proteins. Considering both economical and hygienic aspects, rabbits are attractive for the mammary gland specific expression of recombinant proteins. Using an appropriate promoter, a number of recombinant proteins have been produced from rabbit milk, including hormones. Bioactive peptides and therapeutic proteins, recombinant human proteins produced by transgenic rabbit include different kinds of proteins. They are the recombinant human alpha glucosidase, first transgenic protein from rabbit milk used to treat Pompey's disease. And here, what is the Pompey's disease? Glycogen storage disorder type 2. And here we can also see Pompey's disease is a fatal muscular disorder caused by lysosomal alpha glucosidase deficiency. Alpha glucosidase from rabbit milk is used to treat 4 human babies genetically deficient in alpha glucosidase. That is alpha glucosidase from rabbit milk to 4 human babies who were genetically deficient in alpha glucosidase at starting doses of 15 or 20 mg per kg body weight, and later at 40 mg per kg body weight. The activity of human alpha glucosidase was shown to be normalized in the muscles of these patients, and their tissue morphology, motor and cardiac functions are dramatically improved. And here you can also see the recombinant proteins that have been produced from the rabbit milk, including the hormones, bioactive peptides, and therapeutic proteins. Recombinant human proteins produced by transgenic rabbit include antithrombin, interleukin 2, tissue plasminogen activator, erythropoietin, insulin-like growth factor, extracellular superoxide dismutase, growth hormone, salmon calcitonin, equine chorionic gonadotropin, nerve growth factor, chymosin, clotting factor number 8, bovine follicle stimulating hormone, protein C, lactoferrin, and the beta interferon. And coming to the applications of the animal bioreactors. Applications of the animal bioreactors. Animal bioreactors have many potential uses. Transgenic animals have been created with increased growth rates and enhanced lean muscle mass, enhanced resistance to diseases, or improved use of dietary phosphorus to lessen the environmental impacts of animal manure. Transgenic poultry, transgenic swine, transgenic goats, and transgenic cattle that generate large quantities of human proteins in the eggs, milk, blood, or urine also have been produced with the goal of using these products as human pharmaceuticals. Human pharmaceuticals proteins include enzymes, clotting factors, albumin, and antibodies, and these are synthesized. The major factor limiting the widespread use of transgenic animals in agriculture production system is their relatively inefficient production rates. Use of transgenic animals to clone large quantities of the gene responsible for a cattle growth hormone, that is bovine somatotropin. These are the different applications of animal bioreactors. Thank you, one and all.